Greetings, my name is Riccardo Spott. I'm a PhD student at the Institute for Infectious Diseases and Infection Control at the University Hospital Jena in Germany. Today I want to tell you about spatial distribution of persistent SARS-CoV-2 mutations, as well as how we utilize mobility data for targeted sampling. In our study, we first investigated the alpha spread in Germany and its federal state of Thuringia. Here we show that the initial sequencing in Germany, as well as in Thuringia, was low, but increased in the following months due to the introduction of the German Corona Surveillance Act on the 19th of January 2021, issued by the German Health Ministry. First, cases of alpha emerged by late December 2020 in Germany, but alpha did not spread to Thuringia until late January 2021. So the alpha spread in Thuringia initially lagged around 2 weeks behind Germany. In the following months then, alpha spread rapidly throughout Germany and Thuringia, and ultimately reached similar proportions in both. We also constructed a phylogenetic time tree using over 64,000 German and over 6,200 Thuringian alpha genomes. Here, the Thuringian alpha genomes, colored in red, are scattered across the whole time tree in between the German samples. So no Thuringian-specific alpha cluster was formed. By annotating this phylogenetic time tree with non-alpha specific mutations, we then identified 9 independently spreading alpha clusters within Thuringia, of which 8 are defined by a spike mutation and 1 by an ORF1b mutation. All these 9 clusters spread mostly regionally confined, with single isolates also spreading to distant districts as well as other federal states. Here we see now an example cluster, S939F, also defined by a spike mutation as indicated by the leading S, and it spread over the course of 4 months, beginning in February and lasting until May 2021. The gradient from white to dark purple here reflects an increasing amount of sequenced isolates per district over all months. So we see the cluster originated in the Eastern Thuringian region and mostly spread in this region over the following months, with single isolates also spreading to central as well as northwestern Thuringia. But we also observed a gap in the cluster spread with these 2 gray districts here in between. So in conclusion, all our 9 clusters had a visible and trackable spread, but we had also a potential sampling bias as indicated by these gaps in the cluster spread. To further investigate this sampling bias and eventually predict the cluster spread, we then utilized movement or mobile service data, provided and analyzed by our cooperation partner. Here, blue districts are districts where we not yet found an isolate belonging to the cluster, while in red districts we already found a cluster sample. Further, the darker blue district is covered, the more mobility it gets from all the cluster affiliated, so the red districts. And as we see, the mobile service data partially indicates already the spread of the cluster, with districts turning darker blue or dark blue before they eventually turn red in the following month, or in the case of the 2 aforementioned districts in between here, they are highlighted in dark blue. So also the mobile surveys data indicates a potential sampling bias here, and thereby can be used as a tool to guide the sampling process. Now that we used it with retrospective data, we also wanted to test it with current lineage in the current pandemic events. Therefore, we set up a pilot experiment to use the approach of mobility-guided sampling with the Omicron sublineage BQ11. This was first isolated on 5th of October 2022 in a northwestern Thuringian community, here indicated by the yellow circle. There, we used the past 2 years mobility data to identify the 8 communities receiving the most mobility from this originating community, indicated by these 8 purple arrows. Subsequently, we then sampled 1 batch from the 7 Thuringian communities identified this way, and found another BQ11 sample on the which was isolated on the 17th of October 2022, here shown by the red circle, in proximity to the originating community. Our randomized sampling of the general surveillance, in contrast, did not find any BQ11 samples in the same period, and only found 4 additional BQ11 samples in the 1-month follow-up period in November 2022. So, in conclusion, we had a comparable alpha spread in Thuringia and Germany. This means that our findings for Thuringia are probably transferable to Germany. We identified 9 independent alpha clusters in Thuringia, which were, to the most part, defined by spike mutation, and all of them lasted for at least 2 and up to 5 months, spreading mostly regionally confined. And lastly, we used mobile service data to indicate a sampling bias in our surveillance, thereby supporting our general surveillance either retrospectively, using it on already collected data to examine the surveillance efficiency, or actively with the mobility-guided sampling approach. Here it helps maximizing the efficiency of scarce surveillance resources. And with this, I'm at the end. I thank you for your attention, and if you have any further questions, feel free to reach out to me. Thank you very much.